So welcome everyone. It's really a pleasure to welcome everyone to this Symposium 4 in the Women's Brain Health Conference series. So today my name is Elizabeth Rideout, although it's that name every day actually. But my name is Elizabeth Rideout and I'm going to be chairing the session today on sex hormones and metabolic regulation. Oh, that's great to start off a Monday morning like that, right? So it's really my pleasure to introduce three fabulous speakers, especially in light of the fact that it's International Women's Day. So today I'm joined by Dr. Susan Howlett, Dr. Julia Zacher, and also by Dr. Sharon Ladyman. So I'm really excited to have these three speakers here today, and I'm sure all of you are as well. So I'm just wanted to start off our session by acknowledging that we work and live and play and participate in a community that is located on the unceded, ceded, and traditional territories of the 203 First Nations, along with 38 Métis chartered communities, each of which possess their own unique traditions and history here on this land that we now refer to as British Columbia. In addition, we acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action, the United Nations Declaration on the Rights of Indigenous Peoples, and the BC Declaration on the Rights of Indigenous Peoples Act. In all of our work and research, we're committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. So today, I'm really fortunate to be joining you from beautiful Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. So with that, I'll just turn it over to Lisa to give you a little bit of an overview on the cluster before we jump right into listening from our speakers.
Hi everyone, my name is Lisa Galea and I'm the lead of the Women's Health Cluster, very proud leader of the Women's Health Cluster, but I can't do it without you. We're really growing, so I think we went from 20 to now in 2 years over 230 members and all over the world, so really pleased with that. And it's multi-disciplinary, so it's not meant to just be for medicine, but we also have people in economics and social sciences, humanities, etc. We have a lot of events, especially for trainees. So any of the trainees that are in the audience, or if you have trainees that you'd like to send our way, it's free to join. They have a seminar series, blogs. We're going to have starting up very soon podcasts. And we also, hint, give out trainee travel awards, even though at the moment there's no travel going on, but hopefully with more of us getting vaccinated, there will be some travel soon. We are really grateful. We need to thank our sponsors and individual donors without which we wouldn't be able to present this conference. So we have Michael Smith Foundation for Health Research, CIHR, and of course, Elizabeth Rideout donating a portion of her sex and gender chair. Please do become a member. You can join very easily. It's on our website. We've made it quite easy to join and donate if you feel like you have the resources to do so. So without further ado, I'm going to leave it back to Dr. Elizabeth Rideout to introduce our first speaker.
Thanks so much, Lisa. It's really such a pleasure to work with the Women's Health Research Cluster. The work that everyone does as part of the cluster is so important, especially to recognize it on this International Women's Day. Okay, so I'm going to stop sharing my screen just to allow the first speaker, if they wish to do so, to start sharing their screen while I start the introduction. So today it's really my pleasure. It's actually a massive pleasure. I've been trying to have Dr. Howlett give a talk, and then COVID scuttled the first opportunity that she was going to use to come and talk. So it's really my pleasure to introduce Dr. Susan Howlett as our first speaker today. Dr. Howlett is a professor of pharmacology and geriatric medicine at Dalhousie University, where she runs the Heart Health Lab. Originally from Montreal, Dr. Howlett completed her PhD at Memorial and postdoc fellowship at the University of Alberta before moving all the way to the other coast to Dalhousie in 1989. So she's been continuously funded by CHR and its predecessor, the MRC, throughout her career, which I'm sure all of us can recognize as just an absolutely massive achievement. Dr. Howlett is really well known for her work on male-female differences in the way the heart contracts across the life course, which is the reason I've been very inspired by her work for such a long time. She's got a particular interest in the role of sex steroid hormones in normal heart function and understanding how changes in sex hormones at different ages may predispose individuals toward different cardiovascular diseases. So without further ado, I'd like to introduce Dr. Howlett, who will tell us about the impact of sex steroid hormones on the heart. So please join me in giving a massive welcome to Dr. Howlett today.
Thank you very much, Liz. I really appreciate the generous comments, and thanks very much for the introduction. It's going to be my pleasure to talk here today in this virtual format. I'm really sorry I wasn't able to visit Vancouver. It's one of the great cities of the world. But of course, we're all here, and most of us probably at our houses, but still trying to communicate and share about our work. So I'm going to talk today about a little bit about the impact of sex steroid hormones on the heart. I'm doing quite a lot of different projects on this, so I've tried to take some of the newer things we're doing. I'm happy to talk to people about any of the work that we're doing in the lab at any point, but I will focus on some of the newer things. But I'm going to start out with some of our original work, so more than 10 years ago, and then just lead to the present. Whoops, hang on. Okay, so I guess I'd like to start with the inspiration for why I got interested in looking at sex hormones across the life course. And it was, like a lot of things in science, a bit serendipitous. And it was also probably a good experimental design on our part. So this type of graph has been put out by the American Heart Association and similar publications in Canada for many years, showing things that we pay a lot of attention to, like here, prior to the onset of menopause, men are much more likely to get different types of cardiovascular diseases than women. But if you actually look at this trajectory, and this is just percent of the population with different cardiovascular diseases, you see that the major risk for getting cardiovascular disease really doesn't appear to be sex, it appears to be age, and that the heart disease is happening out here. And so one of the things that inspired the work was, I thought, well, why are we using animals that are more like 20 to 40-year-old people, or even younger, when the disease is actually happening in a completely different place? So that was the first, kind of inspiration for what we started to do. And we asked a really simple question. We wanted to know how age affects the ability of the heart to contract. The other thing is, we thought, well, and this was before, it became quite so popular to use animals of both sexes, which I'm delighted to see is becoming, very popular and has been publicized by things like the NIH. But we thought we would use heart cells from both sexes because we thought there could be an age-sex interaction. And we know that there's receptors for all of these different sex steroid hormones, estrogen, testosterone, and progesterone, on individual cells that make up the working part of the heart. And it's known to, and we'll talk a little bit about the receptors later, but these receptors can have both genomic and non-genomic actions. So estrogen acting on the receptors can do things to affect gene transcription over the long term, but it's also now more recently recognized that there's acute effects of estrogen on these on different receptors in the heart. So the first set of experiments we did, what we did is kind of fun. We took isolated individual heart cells, and we could track their contractions, and with something called an edge detector. So you can see that the cells are getting shorter, and so we know how much they're contracting. We can measure the electrical signals across the cell membrane with electrodes. We can also load the cells with a fluorescent dye that responds to calcium, which is what causes the heart to contract as it gets released and taken back up into internal stores. And so we can use light and measure photons emitted from the cells. And so we can then look at three signals at the same time. We can look at the calcium signal, we can look at the contraction, and then we can look at the electrical signal across the cell membrane. And we can learn a lot about how the heart cells are changing with age by doing this. So I've published quite a lot on this, but this was the very first, or one of the very first findings back in 2010. And this was fascinating. So we look here and we see this is the cell contraction. And we see that the young male heart cells, are contracting the most forcefully, and significantly more so than young female heart cells. But the fascinating thing we saw was that the male cells deteriorated a lot with age. And we didn't see any change in the female cells. And so this led to a whole series of experiments where we looked at how the heart cells handle calcium, and what could be responsible for these changes. So, but we thought this is interesting. So there's a real difference in how the hearts are remodeling or changing with age. We didn't see any change in the contractions in female cells, and we saw this deterioration in cells from males. And so we wondered if things like this could help explain the fact that men, older men, are more likely to get something called heart failure with reduced ejection fraction. This is a type of heart failure where the heart just isn't pumping very forcefully. And maybe it helps explain why older women have preserved ejection fraction. And of course, that's going to be, there's probably many factors involved there, but this was really interesting and consistent with research into heart failure. And some of my current research is actually looking at how these remodeling changes you see in the heart, may promote different types of heart failure in different groups of people, including frail older people of both sexes, older women and older men. However, I would like to emphasize that that female hearts aren't out of the woods for cardiovascular disease. What we saw in addition, and this was fascinating too, was we saw that this is a caffeine transient. If we spritz one of our favorite drugs at a heart cell, it will release its calcium from internal stores, and we can measure that. And what we found was there was a whole lot more calcium stored inside heart cells from these older females than we saw in the males. So there was really no change in any other group. So this is actually not a good thing. This tends to overload the stores inside the heart, or the sarcoplasmic reticulum, with calcium. And it can actually lead to some abnormal activity, as we'll talk about in a second. We have evidence that this involves estrogen because when we overectomize younger animals, we see the similar increase in the stores. So we think that estrogen is involved, and we think that this increased calcium available in heart cells can get released spontaneously. So if you look, this is just some unpublished data that we're following up these now, but these are a bunch of action potentials, which are kind of the normal electrical signal in a heart cell. And we see in older female cells from overectomized females and from aged females that they have these spontaneous releases. And this can actually trigger extra beats and can be responsible for generating arrhythmias. So some of the work we're now looking at is trying to figure out how electrical changes in the heart cells are remodeled in prolonged absence of estrogen. We've also done work in males, and we've looked at gonadectomized males. So these are males that have had, the overectomized are animals that have had their ovaries removed to reduce estrogen and model older people. We've also looked at gonadectomized males, where we've removed the testes, and we see that there's also changes that are going on in individual heart cells there. We have a sham operation versus a gonadectomized animal. And what you can see is this is an action potential. You can see that it gets really long. So the action potentials are prolonged in older males. And this is also a maladaptive age-dependent change. And it's different from what we see in females. And it predisposes towards a different type of arrhythmia. And this event here is called an early after depolarization. And these types of arrhythmias are also life-threatening. So we're actually looking at the channels that are responsible for these changes that we see in male hearts, in older male hearts, in the absence of testosterone. So I hope I've convinced you that sex hormones can affect the ability of the heart to contract, the ability of the heart to store and release calcium, and that it can have impacts on electrical activity in the heart. And there's certainly, there's talks for each of those. There's a lot going on. But we've been interested in the possibility for translating some of the things we've learned about the heart and its response to sex steroid hormones. And so we thought about the fact that age-related heart diseases in both males and females can require cardiac surgery. And when the heart is operated on, it's put on a bypass machine so that the heart is actually arrested, so it stops beating, with something called a cardioplegic solution. So it's pre-treated with this solution, and it's arrested, and then it can experience periods of low blood flow, which can be damaging, and this is called ischemia, followed by reperfusion when you restore the blood flow. And so we thought, I wonder if the cardioplegic solution is effective in older hearts. And also, the second part of this research question was to figure out if there's ways to improve this using some of what we know about these receptors for sex steroid hormones in the heart. So we developed this complicated, it's not really that complicated, but we had to adapt a Langendorf perfusion apparatus. And that's because we had to cool the perfusate, because the cardioplegic solution that they deliver during cardiac surgery is ice cold. And they deliver it for a period of about 6 minutes, and that's what we did. And then we exposed the heart to low, after treating it with the solution, we exposed it to low oxygen conditions for an hour and a half, which is a really long time, but it is trying to model what goes on in cardiac surgery. And what we do is, this is the heart being perfused here, and we put a balloon in the mouse heart, and we're able to measure the force that the heart is developing as pressure. So we can measure pressure signal that the heart is generating. And so what we do is I'll show you in the next slide. We expose the heart. I'm gonna move this thing, okay. We expose the heart under baseline conditions to just a normal buffer, and these are individual beats. They're just, you can see they're quite large. And then we expose it to this cold cardioplegic solution, which arrests the heart, which means it doesn't contract anymore. That's a 6-minute exposure. Then we expose it for 90 minutes to ischemia. And then we reperfuse the heart, and we see if we can recover back the activity to baseline. And what you can see, this is just an example, but in an adult male, you see that there's very little recovery. This was fascinating. In the older male heart, there was quite good recovery. It wasn't 100%, but it was quite good. And the adult female, there was quite good recovery, and it looked even slightly better in the older female in this example. So this is a paradigm that you would see during cardiac surgery with baseline cardioplegia, this long ischemia of an hour and a half, and then reperfusion. Okay. So then what we did is we looked at left ventricular developed pressure. So this is the pressure as a function of time, and this is after exposure to ischemia, and we looked at the recovery. And what's interesting here, if you look at the young and old females, we saw, I think you'd argue that's pretty identical recovery function regardless of age in the female hearts. By contrast, in the males, the worst recovery was seen in the young males. And that was significant. These asterisks mean it's significantly worse than the recovery in the young females. And the most improved player was the older male, with recovery here to 75%. So the highest recovery we saw is in the older male group. And again, we thought this was a really interesting and unexpected result. So we can slice through the heart and we can see the infarcted area, so the area that got damaged by this low blood flow. And that's the white part here that you can see. And I think you can notice that this older male heart really recovered quite nicely. And you can see that the infarct in the older male heart was really small. It was the worst in the younger male heart. And the young and old male female, or pardon me, the young and old females had very similar infarcts. So there's room to improve this cardioplegic solution, which I'll get onto in a second. But it's pretty clear that this older male group is doing really quite well, and the younger male group is not. So we wondered what could be involved, and we measured circulating testosterone levels. And what we found was that there's, as you see in some older men, there's a decline in the testosterone levels in these just generic C57 black 6 mice. So these are the younger adults, and these are the old animals. They're about 2 years old. I should have said that earlier. And these are about 6 to 9 months of age. We don't really measure much testosterone in serum from females. So we see this decline in testosterone. So we thought, I wonder if we gonadectomized some male animals, whether we would see the same improved recovery of function. And this is really fascinating. Again, this is a developed pressure. This is an intact heart, same age animals after gonadectomy. And we see a really good recovery of function. So this is suggesting to us that the low testosterone, it might be the high testosterone is damaging the heart, and that the presence of low testosterone may be protecting the older male hearts. And we saw very small infarcts in these gonadectomized hearts as well. So we thought this was a really interesting result and really not what we were expecting to encounter. So why is this important? Well, chronic exposure to testosterone may actually be promoting injury after cardioplegic arrest. However, if testosterone levels are lower, this might actually be protective in older people who may be, older men who may be more likely to experience cardiac surgery. We also think testosterone supplementation, which has become a bit of a fad, and both men and women are taking it for different conditions like effects on frailty, effects on libido, etc., muscle mass. So we think that it suggests this may be unadvisable in males and females prior to cardiac surgery, because we think that it might augment the risk. And again, these are experiments in mice, but this is a way of thinking about how we could translate these to the clinic. So we know that estrogen has effects on different receptors: estrogen receptor alpha, estrogen receptor beta. These are the nuclear receptors responsible for a lot of the classic things that we think of when we think of estrogen acting on the body. And these are effects that are longer term, so they have genomic actions, and they change the transcription of different genes in tissues, including heart. There's also more recently been a real interest in this GPER. So it's the G protein coupled estrogen receptor. And it's thought to be a membrane-bound receptor. It may be elsewhere as well, but it's thought to mediate acute effects of estrogen. And interestingly, you find all these receptors, but you find GPER in the hearts of both males and females. And so we knew that there's a compound called G1, which is a GPER agonist. And we wondered, could we take this GPER agonist, and these G protein coupled estrogen receptors are thought to be involved in many different beneficial effects in the heart. So we wondered if we could add this to a cardioplegic solution and actually further protect the older hearts from both sexes from ischemic damage during cardiac surgery. And so that we did that, and here's the results from the adult animals. Again, they were about sort of 6 to 9 months of age. And here's the left ventricular developed pressure. In the adult females, here's the control data. But when we added G1, you can see that we got really excellent protection. And I should point out this G1 was added to the cardioplegic solution for 6 minutes. And then all the flow through the heart was arrested, and the heart was exposed to 1.5 hours of ischemia and then reperfusion. So this had a really nice cardioprotective effect. So it's a way of maybe harnessing these beneficial effects of GPER to actually apply it to a clinical scenario. This was interesting too. We saw a tiny benefit in the adult males, but even when we used higher concentrations, we didn't see any increase or much increase in the recovery of function in the adult male heart. We might suggest then that this is because the testosterone is adversely affecting the hearts in adult males. And we saw equivalent data when we looked at the infarcts, and just that the infarcts were reduced by G1 in adult females, and there was no significant effect in adult males. When we looked at the older animals, these are about 2 years old, what we found was G1 was protective in both older females and in older males. And you can see here that the effect looked even better. We got almost 100% recovery in both hearts if we pre-treated with 6 minutes of G1 in this cardioplegic solution. This is a huge benefit. I mean, it's about doubling the recovery in reperfusion. So this is an interesting finding, which we think has potential translational benefit. If you look at the infarcts, again, seeing the white part here, you can see lots more infarct in the control tissues in old males and females. But you can see the infarcts are really very small. And this result here interested us. And so we thought, well, why are we seeing such a benefit in the older males, but no effect in the younger males? And so we thought, well, it can't be testosterone necessarily. But then we realized that the animals get fat as they get older. And there's an enzyme in fat called aromatase that can convert testosterone to estrogen. And so we measured estradiol in the serum from these animals. And we saw no estradiol in the younger serum from the males, but we did see appreciable levels, suggesting that maybe what testosterone they do have is being converted to estrogen, and it might actually be activating potentially these GPER receptors and other beneficial pathways and contributing to this. In the females, we saw no estradiol really in the older animals at all. We saw highly variable estradiol levels in the younger animals. And again, that could be beneficial in those animals, but they don't have the testosterone around to convert. This is a really fascinating paper that was just published in February of 2021 that I found from, I think, Sarah Lindsay's lab. And this was looking at GPER receptor expression in hearts from males and females. And I thought this was really interesting. There was an age and sex-dependent effect, but in both the females and in the males, GPER receptor expression was increased with age. Again, so it's suggesting this might be a really interesting target to use with some of these cardioprotective GPER agonists, for example. So I think, what we think is we've developed a preclinical model where we could test these solutions for use in cardiac surgery, and we think you could add an acute estrogen receptor agonist to this cardioplegic solution when you arrest the heart. And at least in animal models, we found reduced infarct size and better functional outcomes in the aging heart. And given that older adults are more likely to undergo things like cardiac surgery, we think this is an interesting approach that we can use. So in conclusion, I hope I'm not too late here, but age-dependent cardiac remodeling differs appreciably between the sexes, more so than we might have thought. And we think this could help explain differences in susceptibility to different heart diseases that happen as both men and women get older and are subject to different hormonal changes in the background. And we do think the sex steroid hormones are implicated, and they are implicated. There's vascular effects, which I haven't talked about, but I've talked about effects directly on the heart cells. And we think we may be able to harness some beneficial effects of the GPER agonists to improve outcomes after cardiac surgery. So I'd like to thank, that's interesting. The picture of people in my lab went off to the side for reasons I don't understand, but these are current lab members and people who were in the lab that contributed to some of this work, and including Angelique Meyer, who is a great MSc student who's now left my lab, sadly, but she did a lot of the translational work on cardioplegia, and some of my collaborators at different places across the country, and of course, support from both the CIHR and Heart and Stroke. And I'd love to answer questions.
Thank you so much, Dr. Hallett. That was really wonderful. So I think if people have questions, please either raise your hand or put the questions in the chat, and then I can call on you to unmute yourself. So we'll wait to see if I have any. I have some questions. I'm really, I guess I'll take this opportunity while people are typing and unmuting themselves and raising their hands. So I was really curious, the males, the really strong contractility of the heart in young males, do you think there's just like a massive requirement for things like ATP or something like that? And so they really, the kind of impact of that low oxygen is really just being felt more strongly, I guess, in young males than in other groups.
So yeah, I hadn't thought about that particular aspect. I mean, that's an interesting thought on it. We have actually probed into the calcium handling that happens and how that differs between young males and young females. And what we found is, this is pretty interesting, that the ryanodine receptors, which are the receptors that are on the sarcoplasmic reticulum where the calcium is stored, they actually appear to be functioning differently in males and females. The phosphorylation status is different. Oh. And the actual size of what's called a calcium spark, which is kind of a little tiny subunit of the whole calcium release event, those are actually bigger in males than in females. And we actually think that estrogen may be damping down the size of those in females more so than in males. So it could be that there's more calcium and more calcium overload, right, in males. So they're contracting, very forcefully. But in the younger males, the ryanodine receptors may be just in a state where they're more likely to fire off. But that's just a thought. We have shown some of that. So there's definitely differences that happen. And then as the animals get older, we find that the calcium current goes down. And it's probably the case that the female cells are adapting by increasing the stored calcium.
Okay.
Whereas the male cells just start to give smaller and smaller and smaller responses. So that's the way we've looked at it. We haven't done the energetics of it, which would be fascinating and could really be playing. It's probably multifactorial, right? You know, we focused on
yeah.
the excitation, contraction, coupling part that, you know, we've mostly dug into, but there's so many different factors here that we could look at. Yeah.
Absolutely. That's fascinating. So I'm just going to have a look at the chat, and I'm going to keep my eye out for people's hands up. Do we have other questions? Do you want to raise your hand, or? That's great. So we have one question. Pooja, did you want to unmute yourself and ask your question?
Hi, that's a nice talk, Susan. So I was wondering, like, you said that you did the gonadectomy and then said that the testosterone is lower and then it helps in rescue. So I was wondering, like, did you supplement this testosterone in females, like young and adult females, and see any rescue difference or not?
That's a really interesting idea for an experiment, and certainly one that we're hoping to do. We ran into a small snag with some of our testosterone supply because they used to supply it in pellets, and they've, even before the pandemic and everything, we stopped being able to get it from the United States because it's a controlled substance. So we've actually switched to doing more experiments right now on selective androgen receptor modulators. And so we're kind of interested in that possibility, but testosterone supplementation still remains something we're really keen on doing as well. And so, you know, that's going to involve some licensing, and we changed some of our research priorities in view of the pandemic from what we wanted to do to what we could do, as I'm sure everybody on the call, you know, has probably done. So, so yeah, I'd like to do that. We haven't tried it yet, and we should, and are planning to.
That's wonderful. So I think just in the interest of time, we'll keep going, and I'll introduce our next speaker, but please join me again in thanking Dr. Hallett for such a great talk. I really enjoyed that. That was fabulous.
Thank you.
Okay, great. So our next speaker today is Dr. Julia Zacher. And Dr. Zacher is a psychiatrist and neuroscientist. And right now she leads the Minerva Research Group, Emotion Neuroimaging, EGG for short, lab at the Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig, Germany. She obtained her MD and PhD degree from the Medical University of Vienna in 2007 and was a postdoc at the Center of Addiction and Mental Health at U of T until 2009. So her lab really studies how endogenous hormone changes affect the human brain and behavior, and really tries to understand the unique vulnerabilities of women to neurodegenerative diseases, things like depression and dementia. So I think Dr. Zacher's talk today, because of the time difference, is recorded, but they're here you know, in person to answer questions later. So please join me in welcoming Dr. Julia Zacher to our symposium today.
And I'm not Dr. Julia Zacher, but you can see the video, I hope. Yes?
I can see, yeah.
Okay, great. I'm going to play it.
Thank you so much, Lisa, for inviting me to speak today and for initiating this great lecture series in the midst of a really challenging time. In my presentation, I will speak about metabolic risk, endogenous ovarian hormone states, and brain structural networks, and why the interaction of visceral adipose tissue and sex hormones may be critical to understanding individual brain aging trajectories. But first of all, let me provide a bit of background why looking at brain health in this context is important to both sexes and to women specifically. We see numbers of patients suffering from dementia continuing to rise, with predictions of them surpassing 130 million worldwide by 2050. And women will make up twice as many of those patients compared to men. These numbers cannot be explained by the longer life expectancy of women alone. Using non-invasive magnetic resonance imaging techniques, we can visualize structural gray matter networks. Accumulating data in this field show that those brain structural network changes seem to be functionally highly relevant. We see an association of structural brain network patterns with memory performance. And we also see that these regional brain network patterns are also the first to reveal accelerated brain atrophy in patients with a neurodegenerative disease. In addition, it's becoming clearer and clearer that obesity is playing a critical role in how our brain ages and how well we maintain cognitive function as we age. Now, despite clear sex differences, both in obesity and in neurodegenerative diseases such as dementia and depression, most big neuroimaging studies investigating these brain structural network changes in the context of obesity, they have not yet made sex differences or sex hormone state the primary outcome in their analyses. To address this gap, we need more integrative approaches. We need to look at measures of brain structure, cognitive performance, obesity, and sex hormones throughout the entire lifespan and do sex-specific analysis. Such an approach could help to identify critical periods before a disease-susceptible state tips into an irreversible pathology. Now, most studies looking at obesity and brain structure either compare patients and age-matched healthy individuals, so they focus on later life, as this is when symptoms of dementia typically become apparent. Or they focus on young adulthood, often recruiting student samples. And while all of those studies are important, what is often missing is the midlife period. It is challenging to convince humans to spend several hours in an MRI scanner during a busy workday when they're often also juggling parenthood and sometimes even additional care responsibilities. But it is also really important to get those data because we now know that metabolic abnormalities and even first neurodegenerative processes begin 20, sometimes 30 years before we see the manifestation of severe symptoms in a patient. We had the great opportunity to do something like this in a study called LIFE in Leipzig, here in Germany. LIFE is a population-based study which focuses on civilization-associated disease, so dementia, depression, diabetes, and cardiovascular disease. In this study, our participants cover an age range from about 20 to 80 years old. In the first period of data acquisition, we were able to assess more than 10,000 individuals. And for 1,000 of them, we now have detailed neuropsychological testing, MR imaging of both brains and bellies, and an extensive blood biobank. For the analysis I'm going to present here, we are now looking at data from approximately 1,000 healthy participants. We're looking at MRIs of their brains, of their abdominal fat, and we're looking at their peripheral sex hormone levels. Now, I'm sure many of you are familiar with what here's an excellent view of a structural MR image of the brain, like this one of a patient with medial temporal atrophy, which is a process we often see in brain MRIs of patients with dementia. What you may be not as familiar with is looking at abdominal MRIs. The colors you see here, red and blue, code for different compartments of where the adipose tissue accumulates. So in blue, you see subcutaneous fat, and in red, you see visceral fat. So why do we do this instead of looking at a measure which is much more easily accessible, for instance, body mass index? Well, here you can see abdominal MRIs of 2 men in the top row and 2 women in the bottom. They are of similar age and similar BMIs, but they display quite different ratios of visceral to subcutaneous fat tissue. Now, if we believe that visceral fat is a risk factor for brain atrophy, those individuals may have dramatically different brain aging trajectories. But let's take a look first on the reasons why visceral fat may be so problematic for how our brains maintain healthy structure and function. We know that visceral adipose tissue poses a significant vascular and metabolic risk factor by, for instance, promoting insulin resistance, low-grade inflammation, and by sustaining HPA axis dysregulation. But visceral adipose tissue has also been associated with increased lesion load in white matter in stroke patients. It's been shown to parallel higher rates of cerebral vessel disease, increased microstructural brain damage, and decreased hippocampal volume, as well as more severe cortical thinning in prefrontal brain areas, both indicators of brain atrophy, and an overall decrease in total brain volume. And in this context, we are now also looking at sex hormone levels. We were mostly interested in estradiol based in work in animals and some human work suggesting that estradiol can decrease inflammation and support HPA axis regulation. Furthermore, antioxidative and anti-apoptotic properties of estradiol have been demonstrated. We know that estradiol can increase vasodilatation and it can strengthen myelinization processes. So there are several established mechanisms by which endogenous estradiol can act neuroprotective. First, when looking at our data, we had to establish a reliable method to quantify our visceral fat. And we spent quite some time segmenting belly MRIs until we were able to show really good radar reliability. Then we plotted the accumulation of visceral fat across the lifespan. So here you see in blue the men and in red the women. And we see, in my opinion, two different signals that are interesting. We see sex differences, not surprisingly. We know that men tend to accumulate visceral fat earlier than women. And when we look at the red curve, we see an inflection point at the age of 47, which is right around the time when the majority of women start experiencing perimenopause. Now, for the brain imaging data, my graduate student Rachel Cito and our team looked at 3 different imaging modalities. We quantified gray matter volume, cortical thickness, and cortical surface area. And then we combined all 3 of them in a flexible independent component analysis. Using this approach, we then identified a brain structural network, which has previously been shown to specifically reflect intracortical myelin. This brain structural network has also been demonstrated to be specifically vulnerable and show early pattern changes in patients with dementia. Furthermore, pattern changes in this brain structural network have functional relevance as they seem to correlate with memory performance. As a first step, we checked this correlation in our data set of healthy volunteers. And here you can see that we were indeed able to replicate this effect. You see that the more maintained the structured brain network integrity, the better the memory performance of our participants. Next, we here looked at the relationship of visceral fat and brain structural network covariance. And in this, we see an inverse relationship. We see visceral adipose tissue to seem to accelerate brain structural changes, and we see a significant interaction for sex. The slope is steeper in men, and women, at least in this plot, seem to be slightly better off. So what could be protecting the female brains from the belly fat? And this is where we brought estradiol into the picture. We observed a positive correlation of more preserved brain structural network patterns and estradiol levels in the female study participants only, which could indicate a potential mitigating effect, protecting female brains from the negative effects of visceral adipose tissue. To further test this, we performed a moderation analysis. And we found that indeed estradiol levels significantly moderated the negative effect of visceral adipose tissue on brain structure in women. Now, when we're trying to put all of this together, what we're seeing are sex differences in how we accumulate belly fat throughout our lifespans. Initially, men are at a higher risk, but then we see an inflection point at the age of 47 for women, right around when perimenopause starts. We also see these sex differences when looking at the relationship of belly fat and brain structure. And while we see a negative association of visceral adipose tissue and brain structural network covariance in all participants, women seem a bit better protected. Now, this looks like really good news for women's brain health at first. Endogenous estradiol levels seem to protect our brains from the harmful effects of visceral adipose tissue. However, at a second look, it seems that right when visceral adipose tissue starts to accumulate at a higher rate, we also stop losing the endogenous estradiol levels. So, in summary, we here provide additional evidence for the conceptual idea that the perimenopausal transition can be viewed as a window of increased risk for cognitive health. We know that with the loss of ovarian function, we see many subsequent effects outside of the ovaries, and that includes the brain. So when we're looking a bit closer at this critical time window in a purely exploratory fashion, we are here plotting women in the age range of 35 to 55. And we are matching these women both for age and for their visceral fat ratio. For their estradiol levels, we did a median split. So we're contrasting low and high estradiol states. In red, you see high estradiol state. In blue, you see low estradiol state. And the individuals with higher estradiol levels showed significantly more preserved brain structural network covariance, and they also showed significantly better cognitive performance in our memory tests. Now, I would like to emphasize the exploratory fashion of this analysis, and I compared to the 1,000 data points I showed you in the beginning. In this analysis, the sample size was only 82. I would also like to acknowledge the limitations that come with analyzing a cross-sectional data set. We are looking at mirror associations, and of course we cannot infer causation. Now, currently, COVID has unfortunately put a stop to our data acquisition. We have longitudinal follow-up that is underway, and we hope that we can pick up scanning again once everybody's vaccinated. As a next step, we also plan a longitudinal perimenopausal cohort where we do an in-depth characterization of the individual hormonal levels at several time points throughout the perimenopausal transition. But I think what is fair to say at this point is that there may be a lot to be gained from regular screening of metabolic risk factors and screening of ovarian hormone profiles in women during midlife. On a much bigger scale, our data lend further support to the idea of the perimenopause as a neurological transition state of importance, and to the observation that the effects of ovarian aging go far beyond chronobiological aging. This just emphasizes the line of work many of you have been contributing to throughout the years and the potential impact we may have if we keep asking these questions and continue to shine the spotlight on hormonal transition phases throughout the female lifespan. And this includes arguing for why all female samples are sometimes warranted and why sex-specific analysis is important. And with this, I would like to thank my incredible team of undergraduate and graduate students here in Leipzig, in particular Rachel Sido, Matthias Heinrich, and Ulrike Schara. I would like to thank our collaborators and all of our funding sources, and of course, you for your attention. And I'm really happy to now discuss these data and take your questions live. Thank you very much.
Thank you so much for a wonderful talk. Please join me in sort of giving a big thank you. I really enjoyed that. Just continuing on this theme of, metabolic regulation and how important it is for women's health. I think it's just fantastic to see all the different realms that is important in cardiovascular disease, neurodegenerative diseases. So I'll just go ahead. So feel free to put your questions in the chat. I think now I can see questions from everyone now that the chat is public. So Kristen has a talk. Can I just ask you to unmute yourself and ask your question?
Oh, sure. It wasn't a question, just a comment. Our mouse studies find similar findings to what she's finding in humans, and I'm just was incredibly excited to hear the talk and to follow more work coming out of her lab. So,
well, thank you so much, and likewise. You know, we always love to see some of the effects we see reflected in animal models as well. So I'll keep looking out for your papers to cite. Thanks.
thank you so much for sharing your story.
Thank you.
Lisa, you have a question?
Yes, I do. Thank you so much, Julia. Sorry, because you were talking, I laughed at something you said, and then I realized, of course, I'm unmuted, so everybody could hear me laughing. But anyway, it was.
I think it's well deserved. It did pull a couple of funny faces there.
It was fantastic, as usual. I'm just wondering about, and I made, of course, a joke on Twitter about how my belly fat might be protecting my brain, and so, which is not really true, I know, but it
I'm
made
sorry.
me chuckle. I'm wondering, in terms of the really interesting, of course, looking at estradiol, but the estrogens that come from adipose tissue, if I'm not mistaken, often estrone. So I'm wondering if you're thinking about looking at the other estrogens too, and how they might, some of the work have done before in the past has shown estradiol may be more beneficial, estrone might be worse or detrimental. So I'm just wondering.
Right. Yeah. I mean, yes, that's really interesting. We haven't done that yet. We're definitely planning to do that. I mean, you know, you also pointed out, right, the more visceral fat this is also producing, right, endogenous estradiol in different forms. I mean, we've discussed this, and for this particular data set, we just decided that in relation to the endogenous estradiol levels that these women still had from their ovaries, it would probably be a really, really tiny effect. But regardless, I mean, tiny effects can be very important, and it's definitely something to look for in the future. Yeah.
I can see that Stephanie Kobola also has a hand raised. Do you want to go ahead and ask your question?
Hi, thank you. Great
Hi.
talk. I really enjoyed it. And I will say, I also have some exciting animal data that goes along well with
yes, perfect.
So I'm ready to submit, so hopefully it'll be out soon. But I'm curious if you have collected data in your previous cohorts or plan to collect data in your future perimenopausal women about their history of hormone therapy use, and whether you plan to look at if type of hormone therapy, you know, estrogen only or estrogen plus progestin might interact with some of the outcomes that you see.
Right, that's a really good question. And for the data set that I've been presenting here, I mean, in terms of hormone replacement therapy, we did check, and those subjects were actually excluded. What we did not exclude were women who were on oral contraceptives, right, throughout some years of their lives. And we had that information. So we did the analysis with them and without them, and didn't seem to change the, you know, association that much.
Interesting. Thank you so much. Really enjoyed your talk.
Thanks.
Okay, I think we have a question as well from Susan, and then we'll, after that, we'll save the rest of the questions for the Q&A session.
Absolutely.
Sure, thank you. That was a really fascinating talk. So I wonder, this is a little off the beaten path, but a lot of the other work that I do involves frailty and its impact on cardiovascular system and other systems in men and women as they age. And there's definitely data that suggest that, you know, with the given, you know, sort of brain function, the frail individuals of both sexes will be the ones to express more signs of dementia. So I guess what I'm wondering is have you thought about that, like, you know, in any way? And do the people with the higher visceral fat have higher levels of, you know, I mean, pro-inflammatory markers, possibly frailty, or is that something you're thinking about? Thanks.
Yeah, that's a great question. Yes, a definite yes to the pro-inflammatory markers. Here in Leipzig, there's a huge focus on obesity research. So there's definitely a lot of people who are looking into this population-based database and exploring those associations. For frailty, I don't think we've done it yet, but it's a really interesting thing to incorporate in the future.
Great. I'm happy to talk to you about it at any point, but that's fascinating. Thank you.
Thanks.
Thanks so much to everyone who asked the questions. That was a fabulous talk, Julia. That was wonderful. Okay,
Thank
so
you.
we'll go ahead and move on to our last speaker today in the session. It's Dr. Sharon Ladyman, who's joining us from quite far away from New Zealand. So thank you again for joining us at a very difficult time of the morning for you. But anyway, so Dr. Ladyman is a senior research fellow in the Center for Neuroendocrinology at the University of Otago. So she completed her PhD at the University of Otago first, and then worked as a postdoc at the Oregon Health and Science University, pretty close to us actually. So this kind of would have been a return to the Pacific Northwest had we been doing this in person, before returning to New Zealand to take up a position. So she now has a senior position in Professor Dave Grattan's laboratory, which focuses on hormone-induced adaptations of the maternal brain. So in particular, her area of expertise is the neuroendocrine regulation of body weight, appetite, and glucose homeostasis during pregnancy, where her work really focuses on the mechanisms underlying the altered hypothalamic response to satiety hormones during pregnancy that are then associated with increased hyperphagia and decreased voluntary physical activity. Since having children, she has also become interested in the long-term impact. I love this. Long-term impact of pregnancy and lactation on maternal body weight and physical activity. So yes, I'm going to be definitely listening out for that as well. Her favorite hormone is leptin. I'm right there with you, although insulin is probably, probably tops leptin for me, insulin. But her current research can't seem to escape from the hormone prolactin. So I'm really looking forward to your talk today. Again, just fleshing out that trio of talks dedicated to women's health and understanding, and why understanding, metabolism and metabolic changes in women is so critical to understanding health outcomes.
All right, so let me just get this sorted. So first, I'd just like to thank the organisers for inviting me to talk about some of my work. And for the most part, my work has really been looking at understanding energy homeostasis during pregnancy. But we've always had the question of, well, what happens after pregnancy and lactation? And so the work I'm going to talk about today is really focused on some of our early work, trying to answer the question of, do the metabolic adaptations of pregnancy and lactation lead to persistent changes in the regulation of energy balance? And so we know that reproductive experience can lead to long-term changes in many different systems within the body. And I'm going to use this term reproductive experience quite a lot. And what I mean by that is going through pregnancy and lactation, giving birth, and basically becoming a mother. And there was a review from Professor Bob Bridges in 2016, and that's where this diagram comes from, really just trying to highlight all the systems that change once you go through pregnancy and lactation. But of course, I've always been interested in the changes in energy balance during pregnancy. We know that pregnancy is a really demanding state, and so is lactation in terms of the mother's energy and needing to fuel the fetus for development, and also the energetic demands of milk production during lactation. So I was really interested in knowing what are the long-term consequences of reproductive experience on energy homeostasis. So what we know in humans is that in women, pregnancy commonly leads to long-term weight retention. And I think it's about a year after giving birth, many women weigh more than what they did pre-pregnancy. And what we also know from human work is that pregnancy is a significant risk factor for women developing obesity and metabolic syndrome in later life. And one of the key questions for for this increased risk factor is what is driving this? Is this mainly to do with lifestyle changes? We know that becoming a parent can have a massive effect on your lifestyle, and maybe you don't have time to exercise and eat healthy food. Or is some of this due to changes in the biological systems regulating energy homeostasis? And this is a really hard thing to tease apart from work done in humans. And so all of my work is really focused on the mouse model, just using that to try and understand the impact of biological changes and taking away lifestyle, the effect of lifestyle changes that happen in humans. So the aim of this work is to determine which, if any, aspect of homeostasis are affected long-term following reproductive experience. And I'm going to talk about 3 different studies we did. First, we just wanted to look at body weight and food intake after going through reproductive experience and look to see if there's any changes in those aspects of energy balance. Secondly, we wanted to look at leptin sensitivity. Now, we know that during pregnancy, many of the hormones involved in regulating energy homeostasis undergo changes in sensitivity. Mostly the brain becomes insensitive to these hormones. And we specifically wanted to know what happens to leptin sensitivity after pregnancy and lactation. And thirdly, we wanted to know if going through pregnancy and lactation could lead to a susceptibility to the negative consequences of a high-fat diet. So basically thinking pregnancy and lactation, a massive challenge to the body, and then we're going to give them that second hit of a high-fat diet and see if, due to having gone through that big challenge of meeting the demands of pregnancy and lactation, makes them more susceptible to the negative consequences of a high-fat diet. So to start with, this is what we did first. We really just wanted to get an idea of what are the body weight changes post weaning, and this is in the mothers, not in the pups. So this is, we had 2 groups. We have our reproductively experienced group. This is their body weight every week throughout pregnancy and lactation. And for all the studies that I'm talking about today, our control group is an age matched mice that have never been pregnant. And that's this black line here. And so when we look at their body weight after the pups have been weaned and removed to the cages, and for as long as we measure, the reproductively experienced mice are significantly heavier than the never pregnant group. And we've done this out to 3 or 4 months, but our colleagues, Dr. Jose Donato in Brazil, he's taken it out 6 months and beyond, and they maintain a higher body weight than the control group. So we also wanted to assess their feeding behavior at this time, asking the question, do they continue to eat more? And we used our Promethean metabolic and behavioral phenotyping cages to really try to look at the patterns of eating as well as the total food intake. But we saw no difference in food intake at 2 different time points. And we saw no significant difference in things like meal size, meal duration, meal number. Some of these things changed during pregnancy and lactation, but by this time post-weaning, they are back to being the same as those control mice. We can measure a whole lot of different things in these cages, and one of the things that showed a significant difference between reproductively experienced and controls was physical activity. So this graph, these two graphs really just show how much the animals are walking around the cage during a 24-hour period. So here on the left is how much they walk each hour. And you can see in the dark phase, this gray box, they're all walking more, as you would expect. Animals are more active in the dark phase. But you can clearly see that the reproductively experienced animals are less active than the controls. And this on the right, this graph just really summarizes what's going on in the light and the dark phase. And you can see in both those phases, the animals are less active than the age match controls. And so what this data really shows us is two things, is that one, body weight remains at a higher than our virgin controls. So there might be some weight retention from pregnancy that even in the setting where with a mouse, they're not going back to their pre-pregnancy weight, and they have a decreased spontaneous physical activity, but no change in food intake. So secondly, we wanted to look at changes in central leptin sensitivity. So in a very simplified version, leptin is an adipose-derived hormone that is secreted and acts in the brain to suppress food intake. It's sort of making this negative feedback loop to keep adipose tissue at a set point. Now, what happens during pregnancy is you actually want to increase your adipose tissue, so you'll get more leptin secretion, and we don't want this suppression of food intake. So what happens during pregnancy is the brain becomes insensitive to leptin, and we think this is to facilitate increased fat storage. And we can show this insensitivity to leptin two ways. First, we can look at feeding the leptin-induced satiety. So if you give a non-pregnant animal a one-off injection of leptin, they will eat less the following 16 hours. But when we do this in pregnancy, we see no effect, showing that they're not responding to leptin. And we can also look at the number of cells that are responding to that one-off injection of leptin. So here we have two coronal brain sections through the medial basal hypothalamus, and we've got the arcuate nucleus and the ventromedial nucleus, two areas we know leptin acts, where leptin acts, and we can detect the cells that leptin has activated by looking at this molecule STAT3, because leptin leads to the phosphorylation of STAT3, and we can do immunohistochemistry for phosphorylated STAT3 and then count the number of cells. We know that during pregnancy there's no effect, no difference in the arcuate nucleus with the number of cells responding to leptin, but in the VMN we see a significant reduction in the number of cells responding to leptin, which may be involved in leptin-induced satiety. So we wanted to know what happens post-pregnancy and lactation with this leptin insensitivity. Does it come back or does it remain, which may contribute to that increased body weight? So we repeated these experiments in controls and reproductively experienced animals, and you can see here, and this is the feeding effect of leptin, that reproductively experienced animals show that suppression of food intake, telling us that leptin sensitivity is returned in the post-weaning stage. And This is the same for our looking at the number of cells that are activated by leptin. If we look at the VMN, where we saw differences in pregnancy, there's no significant difference between the number of cells responding to leptin in reproductively experienced animals compared to controls, telling us that leptin insensitivity during pregnancy is only there in transit and is resumed in the post-weaning phase. So then we went on to do our sort of double hit experiment to put these animals on either a high-fat diet or controlled diet. And we did this approximately a week after they had weaned their pups. And we put them on the diet for approximately 6 weeks. And so we wanted to know, does reproductive experience make a female more susceptible to the negative consequences of further metabolic challenges? And the negative consequences we were interested in assessing were the high-fat diet-induced increase in body weight and high-fat diet-induced glucose intolerance. So first, we weighed these animals every week on the diet, and you can see the red here is our reproductively experienced mums, and the black is our controls, and the solid lines are the groups on the high-fat diet, whereas the dashed lines were on a controlled diet. And you can see that in both our controls and our reproductively experienced animals, there was a significant increase in body weight on the high-fat diet. But what we saw when we compared the effect between the two groups, we saw no significant difference. So the reproductively experienced mums weren't more susceptible to putting on weight on a high-fat diet. We next went on to look at glucose tolerance in these animals. So here we gave them an injection of glucose and then monitored their glucose clearance over the next 2 or 3 hours. You can see in our control groups, we actually saw no difference between the control-fed animals and the high-fat diet animals. And this was not too unexpected. There's a lot of the high-fat diet work is done in males, and they're much more susceptible to these negative consequences than females. So only going with 6 weeks on the diet, we didn't necessarily think we would see a significant effect of glucose tolerance. However, in the reproductively experienced animals, we did see a significant impairment in glucose tolerance in our high-fat diet group, showing that in the reproductively experienced, but not the control animals, there's an increased susceptibility to this particular negative consequence of a high-fat diet. And we were interested in exploring this a little bit further to try and see what the mechanism for this might be. And as I said before, we know that males are more susceptible to the negative effects of high-fat diet than females. Females usually take a lot longer to become obese and to show glucose intolerance on a high-fat diet compared to males. And one of the reasons for this is some work done a couple of years ago now, where this group showed that the levels of estrogen receptor alpha in the arcuate nucleus play a protective role. And this is because it's a protective role in preventing hypothalamic inflammation. And hypothalamic inflammation is one of the mechanisms by which high-fat diet is thought to lead to glucose intolerance. And so what they showed was that in females, they have more ER alpha in the arcuate nucleus, and it makes them less susceptible to the negative effects of high-fat diet. So we thought, well, maybe this is similar to what's going on between our controls and our reproductively experienced mums. We know that going through pregnancy and lactation can affect receptor levels and hormone sensitivity. Other people have shown that. So we thought maybe there's a difference in estrogen receptor alpha in the arcuate nucleus. So we went on to look at this, and here is some immunohistochemistry from controls or reproductively experienced animals, looking at the arcuate nucleus, and all these black dots here represent cells that have stained positive for ER alpha. And when we count those, we actually see no difference in estrogen receptor levels in these animals. And we've also looked by Western blot and shown that once again, there's no significant difference between any of our groups here. So it seems like, at least in terms of the level or the number of cells that display estrogen receptor, there's no difference. So it seems unlikely that this is the a mechanism that is underlying that change in glucose tolerance in our reproductively experienced mums on a high-fat diet. So just in conclusion, what we've found so far is that just thinking about the biological systems, there's an increased body weight that is maintained after reproductive experience. And we think this might be that there's a new set point for the body, or that the mums put on weight and then never lose it is what seems to be happening. They're not putting on weight at a greater rate than controls. They've just got this this sort of new set point. We've also found that the changes in leptin sensitivity are pregnancy specific and not maintained long term. And 3, we found that high-fat diet challenge leads to impaired glucose tolerance after reproductive experience. And our first hypothesis of estrogen receptor levels in the arcuate nucleus doesn't seem to hold out for this, but it might be that there are changes in other tissues as well, such as the pancreas and insulin responsive tissues, which is something we would like to look at in the future. So with that, I'd just like to thank the various people involved in this project, particularly our research assistant Zin, who's done a lot of this animal work, and of course thank our funding bodies for the grants. So thanks.
Thank you so much, Sharon, for such a great talk. I really enjoyed that so much. So do we have questions? We can put them in the chat right now. And then after we have some specific questions for Dr. Ladyman, we'll transition to a Q&A for all speakers. Lisa, you have a question?
Of course, I almost always have a question, but you know, obviously other people should go. Looks like Jim has one too, so I'll let him go. But a very quick one, Sharon. Thank you.
I'm just trying to stop sharing my screen and it doesn't want to do it.
No, I know it's so, you know, sometimes I go up to view option. No, yeah, view option, stop sharing. Maybe we can do it for you.
It just doesn't want to go on the little stop share. Oh, there we go.
Yeah, sometimes that happens. So I'm wondering, and maybe this is, so you did that leptin sensitivity 8 to 9 weeks post weaning, I think, and didn't see any changes. But I'm wondering, like, so then will you look after challenges? But I'm also wondering even later. So in midlife, when you see, you know, maybe some more changes with estrogen signaling and things like that, does that show up? Do you see changes in leptin sensitivity then? Did you look or
well, I haven't looked that far. I'd really love to like push these out much longer. But just, you know, funding and yeah, basically that's what it comes down to. But yeah, I think it would be really interesting to see much further out, you know, past 6 months, whatever, because who knows how that's affecting the system way down the line. And multiple pregnancies as well. That's the other one, you know. I think that there is data suggesting that, you know, the more pregnancies or the higher parity you have, the increase your chances for, you know, metabolic syndrome and that. So, you know, this is one pregnancy and lactation, and it would be, I'm sure the results would be more striking if we did multiple as well.
Wonderful. I think we have another question from Dr. Jim Johnson.
Wonderful talk, and actually wonderful whole symposium. Really enjoyed it. My question is actually kind of a technical one. I might have missed it, but did you do your, when thinking about the differences between glucose intolerance in the males and females, I've come to understand from another colleague who sort of specializes in this, that that can depend on whether the experiments are done at thermoneutrality. I might have missed it, but did you do your experiments in the thermoneutral situation, or are they just like the rest of us regular people do them?
Yeah, just the regular, yeah, absolutely. And yeah, that's a really interesting question. We're really into at the moment doing some, you know, thermoregulation work, and we have animals at thermoneutral at the moment. And pregnancy and lactation are quite different. You know, our pregnancy rates are much lower, and I don't know, it's who knows what we would see.
And another, if I may, a super quick question. I remember in terms of, like, you're focused on the brain, but in terms of the islets and the beta cells, and I know Dr. Rideout is also interested in this. There was a study quite a few years ago that Nemo Achilles group published, showing that multiple pregnancies and hyperglycemia were required to sort of de-differentiate the beta cells in a way. And have you looked at sort of the functional plasticity of the islets and the beta cells before and after the reproductive experience?
I haven't done very much. I've done a little bit of work just looking at you know, beta cells and insulin secretion during pregnancy and the role of prolactin in that. We do have thoughts. I have a colleague who's a beta cell expert. I'm not at all. With a plan to look what is going on in the reproductively experienced animals. And, you know, it would be great in that to add models of multiple pregnancies as well, because, you know, I have a suspicion that it's probably more likely to be the something going on with the pancreas that undergoes massive changes during pregnancy. So you can imagine that it's just multiple pregnancies getting hit over and over again, and then give it a high fat diet and it just, you know, not gonna maybe respond as well. Yeah, it's definitely something I would like to look at, but the pancreas is a bit out of my my comfort zone.
I'll ask a question after Kristen, but I also just want to say that now is the time for if anyone wants to ask questions more broadly to all speakers. So we have two more questions, one from Kristen and one from Julia, but then we'll have questions for all of the speakers as well. And then I think Lisa had a question for everyone to kind of, you know, finish our session off. So Kristen, did you want to ask your question?
Oh, sure. Really great talk. Thanks for sharing. So for your mice, do you know if there's like cognitive impairment that's different with the glucose intolerance that you see?
I have not even thought about looking at that compared to along with that glucose intolerance. That's no, that's a really cool idea though.
Yeah, it's something I see that like correlates. So I'm wondering if it.
Yeah, no, I haven't. I haven't even put those two things together in my brain. So no, we haven't looked, but yeah, that's got some really interesting possibilities.
Your work is great. Thank you.
Thanks.
Julia, did you want to answer or ask your question? Answer your own question. Not really.
No, but I'm happy to ask it. Sharon, I really enjoyed your talk. Thank you so much. That's fascinating data. And it just struck me really like my own experience and the experience of many women who are my patients who have undergone, have experience with gestational diabetes. And throughout the pregnancy, they're, so tightly monitored. And then once they're breastfeeding, I think there's one more test with completely different criteria, and then nobody sees them ever again. So, I mean, I know that you work in animals, but I was wondering, whether you had any thoughts on how that could translate and what we could do differently.
Well, I think I think it should people should be aware that, going through pregnancy and lactation can have these effects. And I think here in New Zealand, we do have more like a 5-year follow-up for women who have had gestational diabetes. I might have that wrong. I've had friends with it, but I didn't have it myself. And I think it's pretty much like 5 years, they test you again. And after that, they're like, no, if you're fine, you're fine. But yeah, I mean, I think it's just something that we all should be aware of, that perhaps women do, have these biological changes that are going to last, and we shouldn't be expected, people should have that expectation that they're not going to go back to what they were, and that's not just due to lifestyle, that's also due to your biological systems.
Right. Thank you.
That's a fantastic point. Lisa, you said you had a question for everyone. I want to make sure that I'm respectful of everyone's time. So I think we're doing quite well. Did you have a question or comment for everyone, I think?
Yeah, I mean, you had a question too, so I don't want to hog, but what occurred to me with all three of your talks was that they're really good examples of why it's important in your model systems, like whatever disease or whatever you're thinking about, to actually appropriately test, sex and age, which is what Susan was talking about and Julia was talking about. But also other, we tend to want to be really reductionist a lot of the times, and neuroscience into other areas. And, I think it's really fantastic. It's really interesting to build these model systems like organoids and things like that. But then we find out that, visceral fat moderates the relationship, and estradiol moderates the relationship, and then reproductive experience modulates the experience or the relationships that we see. And so I'm just wondering if you guys, it gets complicated, but wanted to comment on sort of appropriate model systems for, the work you do, and the importance of looking at both sexes, just something unique to females, like reproductive experience.
Can I comment on that? I mean, I asked the question earlier about frailty, and this was something that we thought about because researchers here at Dalhousie have been instrumental in developing frailty scales that are used in people all over the world. And I had these mice, and they'd be both 24 months of age, males, females, whatever, and some of them would be in horrible shape at 18 months, and others could be older than 24 months and still much more robust. So we developed in the lab a way to actually measure frailty, right? And that has allowed us to consider health span in a mouse in an analogous way, to what you see in people. It's just surprising how much heterogeneity there is in animal aging as you see in people, because the mice are, well, they're basically couch potatoes unless we give them exercise wheels, which we're doing, and that's quite fun. But even within that, like some mice really love to run, others don't. So they're very heterogeneous, right? And so we're trying to find a way to capture that. So we've done this frailty index work so I think that health span piece, you start to see the divergent phenotypes as the animals get older and they have more stresses to the system, right? And then they start to diverge and some of the stresses could be parity, for example, along the lines of what we just heard. So I think that for me, that's what we've tried to do, to come up with models that more closely resemble human aging and all its, different manifestations.
I don't know if any of the other speakers wanted to comment on that, but I agree, Lisa. I mean, one thing that really seems really interesting from this is, you know, we call a certain disease or endpoint, we call it the same thing in men and women, but what seems very apparent is that can be two or multiple different paths to arrive at a phenotype or to arrive at a disease state. And, you know, by simply studying one sex, for example, whether that be predominantly women or the predominantly female animals because of the way some model works, or predominantly male animals because of the way this other model works, we're really not getting the full picture of the heterogeneity underlying disease progression. I work on normal developmental biology and stuff like that as well. But with all of these things, we're just missing out a lot of information when we assume that there's only one path potentially to a disease. So I think that's a really important point that I don't know if the speakers wanted to comment as well on that.
Yeah, I mean, just quickly for the life database, I mean, we've been trying to ask some of these questions and we have this really rich data set. On the other hand, of course, right, in the neurosciences, I'm sure that also pertains to other disciplines, we have this huge reproducibility crisis, right? And a lot of the response to address that is to, oh, you need to, you know, pre-register every detail and you cannot really do anything else, right? And so we ran into a lot of problems also with our students saying like, you know, we have these signals that we don't understand. This is a really good suggestion. Can we look at that? And then of course, yeah, you kind of have to find a balance. We solved it with very specifically declaring what was exploratory and what was pre-registered. But I think that continues to be a challenge.
I just think it's important to acknowledge, mindful of time, but that reproducibility issues are likely because people aren't.
I completely
right. I mean, sex is just one part, right? That's
absolutely, yeah.
Anyway.
Okay, so I'm just going to be mindful of everyone's time and just make sure that we close off by saying a massive thank you to our speakers today, Dr. Howlett, Dr. Zachar, and Dr. Ladyman, who've joined us from quite, you know, large geographical span today, which is enormous. So I just wanted to say thank you to them and thank you so much to everyone for participating. It's your kind of engagement that makes events like these a success. And so hopefully everyone will join us for the next symposium. I really look forward to these symposia myself. So hopefully you'll join us. So thank you so much, everyone, again, for joining us. Lisa, did you have any other final things to say?
No, I don't even know our next one is. It's sometime in April. And so I know I'm such a great organizer. I should have this all on the top of my head, but I don't. Might be the 12th. Don't know. It's on the website. Stay tuned. We'll tweet it. And thank you again. Thank you to our global representation today.
Thank you so much.
Thank you for organizing. You made it really, thank you.
Yeah, it was wonderful to connect with all of you. Thank you, everyone.